sent you some days ago, many days ago, the lecture. With the lecture, I have also sent you two ways. Alright, some videos. I hope you have looked at them, looked at the lecture notes before coming here, because it will make understanding things much, much easier for you. Alright. So gene inheritance, we'll talk about the outcome of this lecture. This topic is hopefully to describe meiosis, mitosis, to understand to understand the concepts of gene traits, phenotype, genotype, to understand the patterns of inheritance, classical and non-classical, and then to be able to draw a family pedigree, family chart. We will do that mostly in the work session, which is going to be obviously after the holiday. And also I will explain probability of inheritance and recombinant frequency. This is going to take perhaps the most part of, of the parts of that lecture, because it does require some explanation. And you will have an exercise about it in the work session. I will put an exercise as well. Okay, so when we start talking, alright. So when we talk about meiosis and mitosis, it means we are talking about the cell cycle. So what is the cell cycle? باش يتم محاضرة مخدي بخيري أتشمسري كأمانة أزانا ليسري أزانا باشتي أزانا أمانة هيني باشتا مربوط ولا مكتب بلام data cell inheritance chromosome non-classical inheritance chromosomal crossing over أمية زيادة كاتك بو أوانا بو تفاصيل كتان. Okay, the cell cycle is a series of events that what happens is that to involve the growth and dividing of a cell to produce two identical daughter cells. Okay? Now, cell division is important. It's how a fertilized egg, it is how chromosomes and genes and traits are passed from one generation to another. This is meiosis. And also, it is how the fertilized egg, in the case of human, formed by the joining of the sperm and egg, it evolves into a normal human being, into an adult. A large number of divisions happen before this, this egg is forming an individual. But also in the adult life, cell division is continued for maintenance, so to replace damaged or dying cells. As you, I'm sure, already know, in human body, some cells, once specialized, so once differentiated, it means they are fully specialized to do their function of sending synaptic signals, doing specific jobs, for example, secreting some hormones. For example, the nerve and muscle cells, they are fully specialized. They don't divide again. We have other cells that divide daily, like blood cells, precursors that are produced by the bone marrow. And we have other cells that in the middle, that they only divide, they are dividing very slow, or they are dividing once required. So we're talking about normal division here. We're not talking about cancer. برسياركم هي ولا موديولاني كوري أجرد كورسي تهم هيج بابتك لموديولا كان باسي كانسر تان بوأكا لتي أوبي Anyway, I'll inquire about this later. It's alright. Alright, ما بس تم كانسر سيل نك كانسر وفلاني كلينيكال كي لاني كانسر سيل باسكا شون تيومر درست alright ثانية كلا موديولا كان أريح ليكين تكاية محاضرة ساعات ينزل دسبيكا أقدر مورك سيشني أمروتان كرد بوا تكاية أما بو خوتان بكن فير بن بيش وخت بين أما نش يعني it's not rocket science. كيش كاتكورن دانيشن بوي سد خوتانيشن لمانستانيشن. Anyway, so for a cell to go through, stop talking. For a cell to go through a division, it must have a preparation. The preparation is the other stages of the cell cycle. So the cell cycle is the events that a dividing cell goes through. So it has the stages of interphase, which includes growth 1, synthesis, and growth 2. And then the division of the, first of all, of the chromosomes, which is called karyokinesis, sorry. Okay? And then this is followed immediately or with it at the last stage by cytokinesis, which is cell division, well, the division of the cytoplasm. Now, G1 includes the phase where the cell is growing in size. It's doing the normal metabolic activities, for example, protein synthesis or transcription translation of most proteins. The number of some organelles increase. Normal cell activities like sending signals, receiving signals, and so on. All of this happens in G1. G1 varies in the length, its length between one cell to another. Now, it's a question. Bodrentien. Fermun Danishan. Beden. ليك شو هاي زانية مدرسة كيف خويتي؟ درسي أو نبو. Okay. So, generally, it is a sort of a long phase of the cell cycle. But the longest phase of the cell cycles, generally, is the S phase. That means the cell takes longer for DNA replication. Okay. Now, once the cell is finished, the G1, it has, we can say, 3 options, depending on the type of the tissue, on what stage is that organism is. Is the tissue already formed, and finished and specialized, or is it still forming? So either it goes into the post-mitotic G0 or G0, it's called, phase, which means that this cell is no longer, it's actually from G1, it's going to G0. When we say it's going, it only means that the cell knows that it shouldn't start DNA replication. Why? Because it's no longer going to divide. So it continues doing the normal metabolic activities of protein synthesis and other things. Okay. This is usually the case for fully differentiated cells. Bochi drink hatuna. Okay. Fully differentiated cells. No. Fully differentiated cells, it means specialized cells.
Ben sich später.
Hello.
Mike'a getir yani. Dönüyor. Bak.
İşte evet bir sağlasın. شي يمكن ما دقيقي هذا.
وعركيتي.
Alright, okay, اگه ایشی نکردن بس این رو بدن diverse cycle میگوید اوش خواهد Alright, بس هیچی مکرد, okay. Fully differentiated cells, you know what is differentiation, yeah? Okay. Or if the cell is still to divide, for example, a certain tissue is still to be formed, so that cell means after it finishes G1, it goes into S phase, it means it starts DNA replication and finishes it and then goes to G2 and then divides and so on. There is another option which we can consider it another option, and we call that the cell is arrested in G1. It means that it remains in G1 waiting for signals from other cells in the tissue, from the body, to see whether it should go into G0, post-mitotic, or it actually continues. This is for differentiated cells that are actually they can still divide when required or in a slow rate. I gave you examples. The phase follows it is DNA replication. You've taken DNA replication with Dr. Haujin, and the main event happens here is DNA replication. So DNA is doubled, which means that sister chromatids with dip are formed here. Okay? Protein synthesis is very limited. All other activities is really limited, but only the things that are related to DNA replication. Okay, you're right. Okay, so of course, apart from some exceptions like the histone protein formation, this happens mostly in, in S phase. G2 similar to G1 in many things. Again, there is a lot of protein synthesis in it, but not as much as G1. Mostly is the preparation for the proteins involved in division. So the arrangement forming more microtubules. Do you know what is microtubules? Yes. You must have taken it in TOB. They are fibrous proteins similar to actin filaments. They are proteins, but they are thicker. They are involved in many processes. One of the important processes they are involved in is separation of chromosomes. So they form the spindle, and so that in preparation for mitosis. You can see here is the phases. As you can see, it shows S phase as the largest. And yeah, generally speaking, S phase is the largest. Sometimes in some cells it could be as almost the same as G1, but G2 is generally shorter than those two. And mitosis, and you can see mitosis involves starting from prophase ending with telophase. Cytokinesis is not part of mitosis actually. It's basically cytoplasm division, which comes, you can see here, this line here, it means that it can actually start with telophase or at the end of telophase. Depends on type of cell, but usually it does start with telophase and then it finishes after the finishing of telophase. Mitosis is then the division of chromosomes at somatic cell when somatic cell division process occurs. And it's important for growth and maintenance. It happens in eukaryotic cells because prokaryotic cells have binary fission, which is different from this process. And of course, we have something called checkpoint proteins that check the DNA at different stages of the cell cycle to make sure. Check many things, but especially DNA, I'm going to talk to you about it. And as I said, cytokinesis follows mitosis to complete the cell division. The stages, as you're familiar with, are prophase, metaphase, anaphase, and telophase. Of course, this is our decision for please put your mobile phones either off or on silent. You should do that before you come into the lecture. Right. أمشتانك أو تريبويز عشكتني إيوانية محاضرة جاية حتى لا work session يشنا بكري بو أوي attention إيوا لا نبري هر لبر أوي لبر أوي تكايا رزي بكري. alright. بين السر أمانة خو خانك بريالي ندا والله من دسك ما بروفيس إنجا أوستي إنجا ميتافيز دسبيكا خانك علاقينية من دايتشي أمي. It's a continuous process. So it doesn't stop after end of prophase. You say, okay, I'm gonna stop now and then. خير بيت؟ بس بزحمة لبريكك ما يقصد لك خير. بريكك تام كده ما يقصد لك. Alright. So واتا إعمليرا شوار سبيسي بدن. شوار فيسي سركي ماندانا وبوتشيا. يعني scientists have put it this way to understand what are the major events that is happening, so that we understand the process of mitosis. All of it. Okay, so the major thing happens here, the formation of spindle from microtubules, the disappearance of the envelope, of the nuclear envelope, the thickening of the chromosomes. In other words, chromatin becomes chromosomes. You do understand this, yeah? They are the same thing, chromatin with N, right? سيشت معناها chromosome. كروماتين هر كروموسومة بلام كشريكي نامينيته ودريجة بيته كدزويلية دي أو كروماتيني بيوتري باشا وكروماتين بدر أو لكاتي إسبيسا كدي إن إيه كدبل أبي ريبليكيشن أبي أو هر دي إن إيه في كاتولا توت السيستر كروماتيدز باشتر أبي نري لكاتي ميتوسيس لبروي شربيتوا أو تشربوناويا وأوي divisionaka بباشي وبركيوها. Alright. So metaphase is when chromosomes line up at the middle of the cell, or you can say the equator or middle of the spindle. Anaphase when the sister chromatids are separated. Once the sister chromatids are separated, they are called chromosomes. طبعا we can now trace sister chromatids on. Okay. And finally, telophase when the chromosomes reach the poles of the cell and the nuclear envelope starts to reforming and the chromosomes start lengthening. This is the diagram of mitosis, sorry. Of course, interphase is not part of mitosis. اللي رأوا أن نسرعوا معناه ونية أن الشيء كلا بشكاني ميتوس. لا، أما بس كيشان بوي بيشان بري جوازي خانك تشيا. يعني أوه. So you can see here that is the can you see properly? So what you can see here is, it's difficult to see the, in the normal microscope to see the chromatin properly. You can see when you use a dye, so they can look at it there. But you can actually using normal compound microscope, you can actually see the chromosomes, you can see the spindle easily. So this is prophase and metaphase, the aligning of the chromosomes, anaphase, the separation, and the telophase. And let's have a look in the general, the true images. So this is a true image of mitosis, but this is in plant cells. Okay? Again, a certain dye is being used for DNA, for chromatin chromosomes. Interphase. Prophase, you can see the thickening of the chromosomes. Metaphase, you see how they're aligned, and the spindle. Anaphase, you can see this is late anaphase, late telophase, early anaphase. As I said, this is because this is a continuous process and it's not like the cell stops at each stage. Anyway, what you see, the line here is the formation of the cell wall. This is a plant cell, and then finally two identical cells are formed. Here is another image with fluorescent microscopy dyes used. Green fluorescent dye used for the spindle, for the microtubules. blue for the chromosomes, and the pink is for kinetochore. Kinetochore is a protein complex which links the chromosomes, the sister chromatids, at the centromere to the microtubule of the spindle. Right. The checkpoints are a group of proteins. Actually, they are kinases. Kinases and their adapter, their helper proteins. What do? They are different complexes of these proteins that they are checkpoint وهو خالي بشكنينن بالسيطرة خالي بشكنينن. What they do is they check many things before at each stage of the cell cycle before allowing it to go to the next stage. The main thing they check is DNA, if there is a DNA damage. What do? If they find the damage, especially in the DNA, they send signals to repair enzymes. Can anybody give me an example of a repair enzyme? Polymerase, well done. Yeah, DNA polymerases, they themselves are enzymes to replicate DNA, the major enzymes. However, they do proofreading and they do repairing if they can. If repair is not possible, depending on the type of and the position of the mutation, they send signal to another group of enzymes, well, major groups, but the major groups are the caspases. These are enzymes responsible for what? For apoptosis. Do you know what is apoptosis? No? Program cell death. Let you be. apoptosis هو كانسر شو نبي نزل من تصوركم يا لنا مرحلة دولة الاشتكان وريدر بالحال apoptosis is programmed cell death. It means that the cell suicides, but it does it in an organized way so that it doesn't make any problems to the other tissues. Apoptosis is a healthy, normal process that happens daily in our bodies. And unless it is too much, when it is too little, it's not normal. If it's too much, also it causes atrophy. However, normally apoptosis is extremely important to get rid of tumor and cancer cells. Anyway, so if everything is okay, then they allow the cell cycle to go on. We have three major checkpoints. One at the end of G1. This is responsible of what? Of many things. To make sure that the proper signals are there, and the nutritions, all the nutrition required, the oxygen, the minerals, all the things are there for proper DNA replication. But most of all, it checks if the DNA is intact. There is no mutation, there is no damage in it, before it allows for replication. Another thing a type of G1 does is controlling of whether a cell is fully differentiated. It shouldn't go into replication again, so it shouldn't divide again after that. Or, the arresting at G1 to decide whether a cell should go into DNA replication and division once required. Anyway, we don't have one in S phase itself, but there is one at the end of G2. Again, the main thing it checks for, in addition to cell size and other things, is DNA replication. It checks for these proteins, check whether DNA replication has gone right. Okay? And then one in the middle of mitosis, particularly in metaphase. Why? Because this is when the aligning of the chromosome كان لو كاتريزبن بالشوازي ريزبن وبالشوازي كاتاتشمنت بين ابسترينا ميتوتيك سبندلا كانوا تاكيدك او بروتينانا تاكيدك او كاينيزانا تاكيدك كامشترو يداوا بو چونك اما اجا باشيرو ندا شيرو ادا كروموسوم كان بيكساني بجيانا نوا یکی لو شتانی کوری اگر نگل دکتر شیکو تصور کم حاضری بیت و دوای اطلاقا یکسر کروموسوم الابنورمال تیسوری کن نگل دکتر شیکو. نه checkpoint كان كاينيزا كان. زين شونا بروف تشيك بوينت كان كاريان أويا زين وكتشوفوا أويا تو خالي مشكلين السيطرة يعني للسيطرة خلق أجريب زين شزام كيشة هي هي تيرورست شزام هي وين وهان هي لكن المهم كسي كداوا كراو لبوليس وكواسيكا أجر كسي داوا كراو بيت مثلا بوليس بدوايا بقري كأيجرن خبرني أنا بوليس أو هاسيري كشتكا تشيك بوينت كان خويان كاينيز جوري كن لكاينيز إنزيم كان وأدابتر بروتين لقليانا كاريان أويا سيل سايكلكا بوستينن تشيكي أوا كان هليك هي يانا نكتشاكي كان أخويان يا سيلكا هين بالن بو أبوبتوسيس خويان أجا هليك هبيك خبر نيرن بو أو إنزيماني و بترسن بو يوشن فاشا Okay, actually there is much more to be said about this, but I don't have time. I need to concentrate on linkage and all that. Meiosis, as you know, is important for sexual reproduction in eukaryotes. And of course, in the case of mitosis, as I said, two identical cells are formed after each cell division. It means when it comes to the number of chromosomes, we start with the diploid cell, we end up with diploid cells. Yeah? You know what is haploid and diploid? Diploid is 2N in all organisms. In human, N is 23. 2N is 46. Okay? So in every somatic cell we have two sets of each chromosomes. Okay? And haploid is the N number. So we have half of the number of chromosomes. Two divisions, meiosis 1, meiosis 2, ends up in forming gamete cells. And it's important, as I said, for maintaining characteristics, traits between generations. Something really important happens, independent assortment and crossing over, which I'm going to explain. There's also an interphase before meiosis to start all of it. There is G1, there is S phase, so there must be replication. And there is G2. G2 is actually much shorter in meiosis, before meiosis. Anyway, so what cells go through meiosis in human? First of all, we have primordial germ cells, PGCs. PGCs are a sort of germ cells, but they are diploid themselves, and they don't go through meiosis themselves, because what happens is that in female, a type of PGCs form oogonia. This is through mitosis. Oogonia form primary oocytes through mitosis. It is only the primary oocytes that go through meiosis to form ova. They call it secondary oocytes, and then it matures into ova or eggs. A similar thing happens with the sperms. Special type of PGCs go through mitosis to form spermatogonia. They go through mitosis to form primary spermatocytes. Primary spermatocytes go through meiosis to form the secondary spermatocytes, which then mature into sperms. And the diagrams here are showing it. Here, this is the primary. It's also a cystic division. It shows some more details here. And it shows which stage is the meiosis stage and when. And here, this is for the sperms as well. Secondary spermatocytes, when they are forming, this is showing it in more details here. Primary spermatocytes going through meiosis, finally forming what is called early spermati, and these are maturing, not dividing, maturing into sperm cells. Okay. Meiosis, during meiosis 1, I want to go through these a little quick, and then I will allow some time for questions. Something happens, meiosis, first of all, separates homologous chromosomes. Okay, this is really important. Each meiosis has prophase, metaphase, anaphase, telophase, and we end up in human with gametes that contain 23, or the N number of chromosomes. At the end of meiosis 1, we have haploid cells. Why? Because we have the half of the number of homologous chromosomes. أجر هاد homologous chromosome كان لهلية كهردو بيرة كمان هبي أو diploid number. كان يومان هبي لهر chromosome homologous كي يكوبي من هبي أو haploid من هي. إما إلى كمان نية بجماري كروماتيد باشا إن number كبهيج شوية بيوندي بجماري كروماتيد نية باشا بويلي كان تكلتشين. زانم جاران زو نظام إحساس لمكتبه كان أو كان هلا يكيان أكريا وقت مايوسيس بفور إن دسبيعك إن جابت الدو إن جابت هيك أنا وغلطة غلط يزور قبري شوار إن مية وجود مية اشتوانية بدو إن دسبيعك كتاي مايوسيس ون يكسر بيت ون إن كتاي مايوسيس توش هر بيت ون إن بو چون كليرة توشين هيا هر كروموسوم homologous chromosome تكاتولد تو sister chromatis أكاش كجيا أبنا وليه فواو جمارك أواتنا. What are homologous chromosomes? Just a reminder again. والله نازلم حزكيتك تعطيكاتوا لا يخوتوا بزاف لا يرسي بزحمة وسيدة. عزيزنا بس يكلم الشوا أو تفجرين مشتي شوا بتراو هو يأهلي أقل من هيك أقل لا أقل لك أقل كراية وأيدين أنجلك لكراية دانا جرسة دانا جرسة والله أول إيش كروي مشتوى راسنا كارنجر أميال إيش كابزانا هو إيش بكاري أنا مشكلة حلوة لا أو درجايش بكرة أجر دات إنسان عزيز نبي ألف عن مكان إيش بيمكن يزحمن أو هي درجة هي زورنا بابا هي كيا سواش ديمون كان هي شتي كوري أكروش شتي وردي زور باش زور بكال والله يوزر أو دفاع Alright, o panka kan ben cevap.
Tabii istilmak.
Okay.
شتێک چاکەکەی ترس.
باشا
هل يقل لحظة أمنية؟
Hello. Hello. All right. إشي نكر هروا صار كم كم هو دار نجتان لمن نتواني كاتزور وبرينا سر homologous chromosomes أويا جتان لي ميستا حاليا إيش نكر شن هي لبيشوا أواني لدوا ويا درنكاتونا أتوان أنا شن زورا أجر بيتان باشترا ورنا بيشوا تألويها مو كرسية ليرش كرسية كيان برحق homologous chromosomes are those chromosomes that each has come from a parent. So for chromosome, in human for instance, chromosome number 1, we have two chromosome number 1. One maternal, one paternal. This is true for all the chromosomes until 22. But when we get to the X and Y, depends whether it's a female or a male. A female gets one X from each parent, again it's the same. A male gets a Y from the father and X from the mom. Okay, so that means they are almost entirely the same. In length, in the genes, they have the same genes in the same position, and everything is almost the same. They differ in one thing, in alleles. So, on chromosome number 1, which is paternal, there could be a gene here, could be the dominant form of that gene. On the maternal one, could be the recessive form of that gene. باشا من برسيارك ثانية كهمان جينا يكي dominant يكي recessive. معناه شيء. شو معناه كذا on a molecular biology level؟ شيء جينا كان شيء.
The dominant one is going to show the phenotype.
I know. I don't mean genetically. I mean molecularly, structurally. What does it mean? يعني شيء. لا لا. نكلوانيا أبي جواز بيه ميتشنك لحالتي نخوشية سندروما بس مرجنية بله بعضی کیوایا ایو بیر بس لنخوشی أكنا وراسطة لحالتي نخوشی كايا بس بزحمة انتباهاً لحالتي نخوشية سندروميك أديرة كايا right. أديرة هنا on a paternal one there could be a gene responsible for the production of an enzyme. That enzyme normally works, is fine, okay? But on the maternal one, that same gene could be mutated in a way, so the sequence is different. In a way that either it is not expressed, the protein is not expressed, so there is a lack of that enzyme which causes a disease to appear, or it causes a faulty enzyme or a faulty protein to be produced. But what about in characteristics like hair color and other things? What does it mean?
For example,
ليلة كألي mutated تك expression هذا أو بأمانة دو قليل روها ماشي. تي أجا أزاي ما بس تشيبا ولا ما كتاب يبنا. The difference is in the sequence. So the gene are the same. There could be a little difference in the sequence. Okay? راستك إلى حالة ميلاني لك أو يا ميلاني برودكشنك إنزيمك بتايبتي بكرسة تايروسينيز بكرسة لميلاني أو آية نبي يا حبي بلا لحالة ترى تنها أو جينا إكسبريشن بروتينك يتري توزي جوازة بو جوازة چونك السيكونسي جينك كم يك عندي جانختي زور تري اعتمادك جوازي هي بلا مش زوركي ليكك لبرو بروتينك ودروس نبي لا أليلك يتري هر أليلك دومينانت بين ريسيسيف بي يعني سابين أليل هموش لي دومينانت وريسيسيف هي جوازة بروتينك كم يا جوازة
باشە؟
چی واکە ئەگەر دوو ریسێسیڤ بێکۆ بن فڵی فینۆتایپەکە بشابن یعنی ئەو پرۆتینە دروستکەن ئەگەر هەردووچیان لەو حاڵەتەیە هەردووچیان کەمێک جیاوازە لە دۆمینەکە ئەی بۆچی هەمان فینۆتایپیان هەیە یعنی لە دۆمینەکە کە دوو ریسێسیڤ بێک
تۆ لێرە مەبەستەکە ئەوەیە کە دۆمینەنتێ هەبێ تەنها ئەو ئەلیلەی وا ئەو کرۆمۆسۆمەی وا ئەلیلە دۆمینەنتەکەی لەسەرە ئەوە کارەکە نایەڵێ ئەمەکە کار بکە تاوانەوە پرسیارێکی زۆر باشە. It's an excellent question. یان نایەڵێ کاریگەریەکەی دەرکەوێت بە گوێرەی فینۆتایپەکە بۆ نمونە ڕەنگی گوڵەکە و ئەوانەی سەکە سێریەکەی باشە؟ بەڵام لە حاڵەتی کە تۆ هەردوو ئەو کرۆمۆسۆمەت وەرگرتووە کە هەردووکی ئەلیلە ریسێسیڤەکەی لەسەرە هەردووکیان کارەکەن هیچ نییە نەهێڵێ کار ئەو ڕێگەی تر دەرەکەوێت مەسەن ئەگەر ڕەنگ بێە ئەو باشە؟ Alright, of course you know that X and Y are also considered homologous chromosomes, but they are different. They are different in everything, in genes, in length, size. All right. This is a paragraph of the human 23 human chromosomes after replication. As you can see, each one is composed of 2 sister chromatids. Okay, but you have chromosome number 1 and another chromosome number 1. Here you keep the 2 sister chromatids. Okay, and you can see they differ in size and of course in types of genes and number of genes. All right. In prophase 1, something really important happened. Jari break when nagging سبكة توزيج كأمامي أو شيء وأمانية توزيع صحي يعني تركيزي أوي أو دايبريك. Right. Bedeng. All right. So we have homologous recombination that happens at prophase 1. In fact, it happens in prophase 1 and part of metaphase 1 as well. Anyway, but it does start in prophase 1. What does it mean? Pieces of DNA are exchanged between homologous chromosomes. Crossing over happens. It is not between the sister chromatids of the same chromosome. It is between homologous chromosomes. And if crossing over occurs between non-homologous chromosomes, that is one type of chromosomal abnormality which I think you will take in the lecture with Dr. Sherko. So that is an abnormality. But this is a normal process that happens all the time and it leads to the appearance of new features. All right. So sometimes it leads to the appearance of diseases, but usually anyway. So please, your attention, I want your attention, full attention now, if you haven't so far. Lera, because here Kurd Nabi Lera, is there anyone who's not a Kurd here or doesn't understand Kurdish here? You don't? Can I? Okay, I'll explain in Kurdish and then in the break I can try it. Okay, right. Lera, chromosomi babla inama chromosomi jmareyaka. Yakam chromosomi jmareyak awayawa yek hatwa le 2 sister chromatids yek yan portaqali yek yan suri. Chromosomi yakakay tiri babla inama maternala paternalaka the other chromosome number 1 yek hatwa le 2 sister chromatids because we are showing after replication before meiosis. Zerdu sauce. Basha? أم دوانا أما هموي وأما بيان أو تري homologous chromosomes أما زور تنفي ساكانة دباري كمان. بصير كيف بزاني نموني دوا سيجين يكيكي بإكس كيكي إكس داينا ويكيكي زد يكيكي واي. هر جينية بابلين ببرسل لشتي بابلين أما ببرسل لرنجي قش أما لرنجي تشاو أبا رنجي بيس طبعا رنجي تشاو ورنجي بيس بيبينين دواي تۆزێ معقدترە لەوە بەڵام من ئێستا بۆ ئاسانکاری ئەم نمونەیەکەم باشە هەر یەکیان ئەوەڵ جارۆک نمونەکە پێشانیەدا X کاپیتاڵەکان واتە دۆمینەنت فۆرمەکان با بڵێن لەسەر مەتێرنەڵە مەتێرنەڵ ڕسێسف فۆرمەکان لەسەر پەتێرنەڵە واتە با بڵێن ڕەنگی قژی ڕەش واتە ئەو گامێتانەی کە هینایسە ئەم کەسە گامێتی دەست بۆ هاتووە لە دایک و لە باوکەوە ئەو هینەی دروست کڕیانی فەرتەلایز ئێگەکە و ئەم گامێتانەی هەیە واتە چی هەیە لە دایکێکەوە با بڵێن کە قژی ڕەشە پێستی تۆخە چاوی تۆخە چاوی ڕەشە باشە و باوکێک کە هەموو شتێک بە پێچەوانەوە قشکاڵ پێستاڵ چاوکاڵ با بڵێن ئۆکێ لەم حاڵەتە با بڵێن با لەسەر مەتێرنەڵە هەیە ئێستا ئەمە بەس ئێستا ئەم ئیندیڤیدوەڵە ئەم گامێتانەی هەیە ئەم کرۆمۆسۆمانەی هەیە ئەم سێ جینە وەکو نمونەت باس لە حاڵەتی crossing over چی ڕوئەدا as you can see pieces of DNA are exchanged لە بینی زەرد و سەوزەکە نا لە بینی پرتەقاڵی و سوورەکە نا بەڵام سەوزەکە لەگەڵ هەر یەک لە پرتەقاڵەکە یا سوورەکە ئەگرێ و بە پێچەوە ئاینی شت زەردەکەش لەگەڵ یا پرتەقاڵی یا سوورەکە ئەگرێ وەبزەمە واضحە so you end up with recombination here واتە لێرە چی دروست دەبێت ئەمانە ئیسە جیا کراونەتەوە مە دوای مایۆسیس تەواو بووە ئەمانە گامێتەکانن هەر گامێتێک یەکێ لەمانی تایە واتە ئەتیمادەکە ئەم کەسە هاوسەرگیری لەگەڵ کێ ئەکا ئەو چ گامێتێکی هەیە ئەگەر هەیە تەوان لێرە ئەم گامێتە بۆ چییە ڕەنگی قژی تۆخ ڕەنگی چاوی کاڵ ڕەنگی پێستی کاڵ بۆ هاکەزە ئەمە ببینە ئیتر ئەم کۆمبینیشنانە هەیە ئەمە کڕۆسینگ ئۆڤەر یەس No, that's what I said. Crossing over ئەمە بزانە ئەو crossing over هیچیش ناگۆڕێن نییە بۆ چونکە ئەمە بزەبت کۆپی ئەمەیە زەرد و سەوزەکە کۆپی یەکن بزەبت واتە ئێکسمۆڵ لێرە هەیە لێرە بزەبت ئێکسمۆڵ هەیە ڕسێسی فۆرمەکە هەیە ئەمە بزەب ئەمە دوای DNA ڕەپلیکیشنە
ئەوە بەتەنها هی مەتەرناڵیە
ئەمە بەتەنیا هی پاتێرنەڵە با بڵێن ئەمە هی مەتێرنەڵە بە پێچەوانە منیش ئەوەم وتگیانەکەم لێرە کرۆسینگ ئۆڤەرەکە بینی ئەم دوانە نییە بەجیا و ئەم دوانە بەجیا نە لە بینی یەکێ لەمانە لەگەڵ یەکێ لەمانە یەکێ لەمانە لەگەڵ یەکێ لەوانە ئەوەیە بەبستەکەم ئەوە کرۆسینگ ئۆڤەرەیە ئەوەی ئەگەر ئێستا تۆ لێرە ئەمە کرۆمۆسۆمی یەکە ئەمەش کرۆمۆسۆمی یەکەکەی ترە ئەگەر مەسەلەن کرۆمۆسۆمی ڕقم یەک لەگەڵ کرۆمۆسۆمی چواردە بیکە هەیە حاڵاتی وا هەیە ئەوە هین دروستەکە سندرۆم دروستەکە سندرۆم هەیە کە ئەو شتانە هینە ترانسلۆکیشن ناوەکەم لەبیرە چێتەوە ترانسلۆکیشنە چییە خۆی ڕاستی تا ئێستا ئەوەی کەشف کراوە بزانم تەنها دوو حاڵەت هەیە کرۆمۆسۆمی 22 چواردەیە شتێکی وایە ئەنی سندرۆم دروستەکە Okay. So don't need to go through too much details of meiosis. چی ڕۆدا ئەینەن پرۆفیسی هەیە هەمان ئەو شتە سەرەکیانەی کە لە مایتۆسیس هەیە لە پرۆفیس ئەو شتە سەرەکیانە بەڵام alignment of chromosomes in the middle of the cell is in a way so that homologous chromosomes get separated ئەو تابینە crossing overish بووە لە بینیانە homologous chromosomes are separated and the two cells that are formed at the end of meiosis 1 are haploid cells and then they go through meiosis 2 where finally the sister chromatids split and you end up in again a haploid number as well so each division ends in formation of four cells haploid cells okay I think you need a break. How about 10 minutes, 15 minutes maximum? شارێک زیاتر نەبێت کایە لەبەر ئێوەمە